Hello everyone, this is a pre-lab lecture for Lab 4, which discusses ion exchange chromatography. So by the end of this lab, you're going to need to be able to describe the principle of ion exchange chromatography, aka IEX. You need to use the PI of protein to determine approximately when and where that protein will dilute, and then explain the role of a salt gradient in ion IEX chromatography. You need to be able to select the appropriate fractions from an elution based on the endpoint assays, which is just a modified activity assay, to be able to use a Bradford assay to quantify protein concentration and then understand the limitations of an assay. You need to be able to explain why we need to dialyze a sample before and after the IEX column. Based on the volume of the sample and the concentration of salt needed for the next purification step, you need to calculate the amount of dialysis buffer needed and whether or not 1 or 2 steps is preferred. And then finally, for this lab and throughout the next lab where we'll be doing affinity chromatography, you need to construct a purification table to track the activity, dilution factor, relative activity, volume, total activity, and percent recovery to track the progress of the purification. And then also be able to explain and rationalize why those numbers change over the course of the purification. So let's start talking about the basics of ion exchange chromatography, or IEX again. This type of chromatography separates proteins based on their charge. So in a protein, I want you to take a minute, or where do proteins get their charge from? I want you to pause and write down some of the places where proteins may get their charge from. All right, so some examples include charged side chains. So you can think of side chains like histidine, arginine, lysine, glutamate, glutamate, and aspartate. So those are some examples of charged side chains. In theory, they can also get their charge from the C terminus as well as the N terminus. And so you have to consider the contributions of all the side chains, as well as the C terminus and the N terminus, and the pH at which the protein is at, to determine what the overall charge of the protein is at a particular pH. And the piece of information that summarizes this knowledge is the PI, or the isoelectric point. And the isoelectric point is simply where the charge of a protein, or the net charge of a protein, equals 0. And so again here, so if we have a protein with a PI of 6, there we go. That means at pH 6, our protein has a net charge of 0. However, if we are at a pH that is less than PI, that protein will carry a positive charge. And if we're at a pH that's greater than the PI, then the protein will carry a negative charge. And this is something that's really important to understand, because you're going to be asked this in questions in the midterm and in the final and throughout the semester. And this is really important for understanding why a certain type of affinity or of ion exchange chromatography is chosen, and whether or not a particular protein will stick to that column depending on the protein that you choose. And so if we're working with LDH, which has a pH of 6, what is the charge during the purification? Okay, so with LDH, there we go, we have a pH of 6, and throughout a purification, at different steps, our protein is going to have a pH anywhere from 7 to 8. Specific buffers are listed in the lab manual, and so we're at a pH that is greater than the PI. So in this case, LDH would be carrying a negative charge. So LDH is going to be negative over the course of the purification. And so for this reason, we're going to want to use a type of ion exchange chromatography to which an anionic protein is going to stick. All right, and so the resin that we use is called a Q-sapphirose resin, and this is a quaternary ammonium resin. It's a very strong anion exchanger. And so if we have an anion exchange column, this means we want a negative column, or not a negative column, excuse me. We want a negative protein, negatively charged protein, to stick to the column. And so if we want a negative protein to stick to the column, that means we want, we would need a positively charged resin. And if we look over here at the structure of Q-sapphirose, again, oh, and that would be a quaternary, we have again a quaternary amine. That would actually be a quaternary amine there. It's positively charged. So again, the positive, so for anion, so if we have the resin and the protein, an anion exchange, the protein is going to be negatively charged and the resin is going to be positively charged. And if we do cation exchange, the resin or the protein is going to be positively charged, so the resin will want to be negatively charged. So how does IEX work in principle? So what happens is at a low salt concentration, so if let's say we have our positive balls here are the resin, so those are our resin beads, and the negatively charged guys there are the protein of interest. So when we have a low concentration, some of the salt molecules are going to solvate and be attracted to the positively charged resin. So for instance, if we have, let me change colors here. If we have NaCl, it's going to be NaCl in our buffer, it's going to be Na plus and Cl minus. Those Cl minus ions are going to be attracted to the positively charged resin. However, when we add high amounts of salt, the chloride ions are actually going to compete with a negatively charged protein to bind to the positively charged resin. And there comes a point where if you add enough salt, depending on how negatively charged the surface of the protein is, it's going to outcompete and kick that protein off the resin. So the salt competes with the protein to bind the column. So if we have a more strongly charged protein, requires more salt. So the more negatively charged the protein, the more chloride ions or the higher concentration of salt is going to be required to kick that protein off. One thing to keep in mind when we're thinking about exchange is that the concentration of our protein is going to be in the low millimolar at the very highest. More likely, we're going to be in micromolar concentrations of our protein. However, the salt is going to be in the hundreds of millimolar, all the way up to 1 to 2 molar. So we have one of the reasons why this is able to work is because the concentration of our salt is going to be several orders of magnitude higher than the concentration of the protein, and that's another way that and the concentration and the small size of the salt relative to the protein is one of the reasons why salt is able to effectively outcompete the protein binding to the resin. All right then. So what does a typical ion exchange run look like? So what happens is, so we start with this column. We'll actually pack the column during lab, and our column contains the positively charged Q anion exchange resin. So first things first, what we have to do is we have to equilibrate our column. So we have to put our column in the buffer that our protein is stored in. We have to put the column in storage buffer. And this generally takes 2 to 4 column volumes of buffer. So if your column is if you have a 5 mil column, that's going to be 10 to 20 mils of buffer. And at this point, you also have to make sure when you're washing the column that the buffer level is always a little bit above the resin, because if you allow the resin to dry out, that will affect the quality of your results and it'll lower your yield, which we don't want. But then once we've rinsed our column and put it in the protein buffer, then we have to actually load the sample onto the column. And so you want to load it, so you want to keep it at all times about a 1/4 inch above the top of the column, and then gradually add more until you run out of sample, and you stop it again about a 1/4 inch above the top of the column. So once we've loaded the sample onto the column, then we have to go and we have to get rid of all the we have to get rid of all the positive proteins, the neutral proteins, and then any proteins that are just non-specifically binding onto the column, or as I like to say, blomming. And this is with a low salt concentration, and this will take about 20 to 25 mils approximately. All right, so yeah, so we have to make sure that we give all of those other junk proteins that we don't want, the ones that aren't negatively charged and going to stick to the column, we want to wash them off. And then and so you'll be tracking the protein content in your run by using a Bradford assay as well as activity assays. And so once you see that the protein concentration is starting to drop off after the wash, then we begin elution using a stepwise gradient. And so what happens is as we progressively increase the concentration of salt, we're going to be increasing the chloride ions, which means we're going to be increasing the competition to bind to the column. So as we increase the salt concentrations, the more negatively charged proteins are going to come off. And again, in this gradient, you're going to be running the activity assays to figure out which of those fractions contains LDH. And this is important because, again, remember that our 75, or rather our 65% of pellet for this lab is a mixture of multiple proteins. And that's why, again, we have to do these multiple steps each time to target a different protein. So here are a theoretical mixture of proteins that are contained in our sample. And based off the PI and the charge at a pH of 8.5, I want you to predict whether or not this buffer or that protein is going to stick to the column. So you can go ahead, pause it, and then check with my answers. Okay, so if you recall, with PI, at a pH that's less than PI, the protein's positively charged. At a pH that's greater than PI, the protein's negatively charged. All right, so let's look at a hypothetical mixture of proteins, because again, all throughout our purification process until theoretically the last step, our mixture is going to contain lots of different proteins that come from a cow heart. Let's take a minute and let's figure out which of these theoretical proteins would stick to the column and which ones wouldn't. So if you want, go ahead and pause the video and give it a try, and then click back when you're ready. All right, so for the first protein, the Pac-Man protein, it has a pH of 5, or a PI of 5. And this PI is less than the pH. So, okay, so if we have, again, let's go back to our little PI chart back up here. If the PI is less than the pH, the protein is going to be negatively charged, and if the PI is greater than the pH, it's going to be positively charged. So just putting that in a different perspective up there, because that can get confusing. But our protein, which has a PI of 5, is going to be negatively charged at a pH of 8.5 because the PI is less than the pH. And so yes, this protein is going to stick to the column. The star protein, which has a PI of 7, that PI is also less than the pH of 8.5, so it too will carry a negative charge and will indeed stick to the column. This protein here has a PI of 8.5, so which is exactly the same as the charge of the buffer. So the charge of this guy is probably going to be neutral and likely won't stick into the column. The purple protein, the PI of 9, now is greater than the charge of, or than the pH of the buffer. So this protein will carry a positive charge. And the same goes for the little ear protein down here. Its PI of 11 is also greater than the pH of the solution. And so these two proteins also won't stick to the column. So it's likely that these three proteins at the bottom will come out in the wash, whereas these two proteins on top are going to come out in the salt. And based on what I said about ion exchange earlier, which of these two proteins, the Pac-Man protein or the star protein, do you think will come off the column first? Well, I would think that the star protein would be loaded first at a lower salt than the Pac-Man protein, and that's because since the Pac-Man protein has a lower PI, it's going to carry a more negative charge at a pH of 8.5, and therefore is going to take more salt to kick it off than the star protein. All right, so here and there, I referenced how you would determine where LDH is coming off, but now we're going to talk about it in full. And so there are actually two different methods that you're going to be using simultaneously as you're collecting fractions to determine where LDH is coming. And the first is the Bradford assay, which we'll talk about a little bit more in a minute. And the Bradford assay tells us the total protein in our sample. And the way that we do that is determining by the micrograms of protein in the well, dividing the micrograms of protein added by the microliters added to that well to get the microgram per microliter or the milligram per milliliter concentration of protein in your sample as a whole. Okay, and so what the Bradford assay does is it tells us how much protein is in each step or is in each fraction. And so the Bradford tells you whether or not there is protein. And this is going to be especially important at your wash step because whether or not you're seeing protein and if that concentration starts to taper off is going to tell you whether or not it's time to continue on to the next step, which would be the salt gradient. So if the Bradford tells us whether or not there's protein, the LDH activity assay, which is similar to what you performed last week and the week before, that LDH activity assay is going to tell us if the protein that we see in our fractions is that LDH. So the Bradford tells us, hey, is there protein? And then the activity tells us, is there LDH within the mix of proteins that are in that well? Okay, and so this is going to be a really kind of crazy lab, and you're going to be collecting fractions, and then as you're collecting those fractions, you're going to be assaying them for the presence of protein and also for the presence of LDH. And so during the wash step, you're not really expecting to find your LDH there. If you find your LDH there, you did something wrong or you overloaded your column. And so since we're not really expecting to find LDH there, your wash step, you're going to be taking 5 mil fractions. And again, what's really helpful is when you get your fraction tubes is to mark approximately where 5 mils is so you can show yourself where to stop collecting the fractions. But then we know that LDH is negatively charged. We suspect it's going to be coming off during the sol gradient. So at that point, you're going to start collecting smaller fractions so you can hone in better on where LDH is. And you're going to be taking elution. You're going to be taking, in the elution, you're going to be taking fractions every 3 mils. So you have smaller fractions, you're going to be collecting them more frequently. Now, when you're going to be collecting these fractions, when you are measuring your Bradford, you're going to want to take a Bradford assay on every fraction. And so with every fraction you get, you're just going to take it and you're going to plop some into your multi-well plate. And it's not necessarily a lot of work. You're just going to be looking for a color change. You're not going to quantify anything with the Bradford until the end. With the activity assay, however, as you might suspect, things get a little more complicated. And in the wash, you're going to be doing it every 5 fractions. And then in the elution or the salt gradient, you'll be doing it every 3 fractions. And so once you have found your active fractions, then you're going to go and you're going to fill in the blanks around those fractions. So you're going to go back and you're going to repeat activity assays on either side, on the fractions on either side of your most active fractions, and then you're going to pool the 3 most active fractions. And I know you guys are probably sweating thinking about running these activity assays every few fractions because if you remember in week 1 or / week 2 was just so much stress to try to get the right dilution and your line perfectly linear. What's different about this week is that you don't need to be linear. At least not initially. You're just looking for the presence of LDH. And so what happens is if you're monitoring you have those orbits of 340 nanometers and you start off with your blank and then you immediately go up, well then you know that you have a change in absorbance of about 2 because you went from 0 to 2 and maxed out the detector in the presence of a minute. And again, you're just looking for the maximum slope here. And you don't necessarily need to be too precise with determining the slope because you just want to know whether or not LDH is there. Once you've filled in the blanks and you've pooled your fractions, which by the way, it's super important to know that you're going to save this for next week, give it to Lindsay. Do not throw this in the trash, but you're actually going to go back and take those 3 most active fractions and you're going to run a full activity assay at the end of class. And that way, you can figure out how much LDH is present. You can estimate how many units of LDH are in your sample at that point. So you're going to check for the presence of LDH throughout your purification. You'll be using the Bradford assay and then a more imprecise activity assay. Once you find where your LDH is, you pull your 3 most active fractions. Then you're going to take a little bit of it and use that to run an actual full-blown activity assay, and then you'll save your fractions, we'll dialyze them, and then next week they're going to go on an affinity column. So let's talk a little bit more about this Bradford assay that I keep measuring. Again, the Bradford assay measures total protein. So in theory, the Bradford reagent, which contains a Kumasi dye, in theory, this dye binds indiscriminately to proteins as long as they contain positive residues or amino acids. such as lysine or arginine, or lysine, sorry guys, and arginine. That's not always the case. Bradford sometimes doesn't like proteins. However, the Bradford assay does work really well for LDH. So, as well as the proteins that are in our lysate. So the Bradford assay binds in theory equally to all proteins, or rather this Kumasi dye that's in the Bradford reagent does. And what happens is when this dye is located on its own in solution, and when you get the Bradford reagent in solution, you're going to notice that it has a brown color. And when you put it in the spectrometer and you look to see if you see an absorbance at 595 nanometers, there will be no absorbance. It would look completely blank, or it'd be pretty close to the background. However, by the way, this is not to scale, this picture down here. Once the Bradford Kumasi dye binds a particular protein, the spectroscopic properties of the dye change and the color of the solution turns to blue. And if you were to put it into the spectrometer and measure it at 595 nanometers, you would see an absorbance at 595 nanometers. So as you increase the concentration of protein in the solution, the absorbance at 595 nanometers is going to increase, and also the intensity of the blue color is going to increase. And so the over a particular range, a concentration range of protein, the concentration of protein is going to be directly proportional to the absorbance at 595 nanometers. It's going to be a linear relationship. And so that's why, I'll explain how we exploit that for the purposes of measurements. And so what we do in order to use a Bradford assay quantitatively is we generate a calibration curve using known amounts of bovine serum albumin, or BSA. And so what we do is we take those standards, and we take a predetermined solution of LDH, and we add BSA to different standards. So we have 0 micrograms, 0.5 microgram, all the way down to 5 micrograms. And so then what we can do is we take our standards that have a known amount of BSA in micrograms, and then we determine the absorbance at 595. And then on the y-axis, so let's say, I don't know, this is 0, this is 0.0 or 0.02, and this guy is 0.5. You're going to graph the absorbance at 595 on the y and the corresponding concentration, or not the concentration, the number of micrograms of BSA in that sample on the x. And then you can use a line, you can do a linear fit to get a calibration curve. So then what happens is when you take your sample, let's say I have a sample and my absorbance of 595 nanometers is 0.3. We go ahead, we fill in the blank, 0.3. That means we're going to correspond to about 1.2 micrograms of protein, and the 5 microliters that we pipetted into the tray. All right. So again, practical aspects of setting up a Bradford assay. You're going to be setting up a 200 or a 96 well plate, and in row A1 to H1 is where you're going to have your BSA standards. All right. And so that's where you'll add your increasing amount of BSA, increasing micrograms. And so what you'll want to do is when you have your 0 microgram sample, you're going to want to add 5 you're just going to want to add, or you want to make sure when you're calculating how much BSA to add, that you're adding a total of 5 microliters of water as well, because when you're making in all the other wells, when you're tracking your fractions, you're going to add 200 microliters of Bradford reagent and then 5 microliters of protein. And so to ensure that all of your wells are the same volume and therefore have the same path length, you want to make sure that you're adding the same amount of volume and the standards as well as in the samples. Now, you're going to use the Bradford assay visually. So depending on the intensity of blue, that's going to tell you about how much protein's in that fraction, but you're not going to read your samples and actually use them quantitatively until the end of class, and we're going to run them on the Prate reader. And again, I'm going to emphasize this. It's really important to keep up with your fractions as they elute. I recommend that you have one lab partner running the column and handling the fractions, and then another lab partner be responsible for keeping up with the Bradford plate, and then also running the activity assays. Okay, so again, just to review, the Bradford assay is going to tell us the total protein. We're going to do a Bradford assay for every single fraction throughout the entire purification process. The activity assay tells us, is LDH present in that fraction? It's going to also help tell us which fractions to pool, so combine together and then add and use for the next step. And for the wash, we're going to be testing every 5 fractions. And then for the gradient or the elution or the salt, we're going to be testing every 3 fractions. And then this is a lot of information that you're going to have to keep track of. So the first thing I recommend you do after we do the problems in class is to set up a Google spreadsheet and your lab notebooks to track all of these quantities. And so the first column, you'll have your fraction number. So 1, 2, 3, 4. And then we'll just do a little there and go 20, 21, 22, 23, 24. In the second column, you'll track your activity. So in theory, all of your activity in those early fractions are going to be 0. And then maybe once you get into your wash, so maybe then your protein concentration will be, your NaCl will also be 0, and then you can backtrack with the Bradford assay to fill that in later. But then once you get into your gradient, which you'll keep track of, and you'll just need to put the concentration of NaCl at the first fraction in each gradient. You may notice a little bit of LDH activity. And then once you hit the particular fraction where LDH is located, and it's not necessarily at 0.8 molar NaCl, you'll see that activity rocket up and then eventually tail down, or I guess it would be 33. And then that's where you'll want to go back and fill in those fractions on either side of the activity that have fractions to help you find which ones are the most active, which ones contain the most LDH. And so in this case, if we were to go back after and say, oh, this guy's 2, this guy's 2, that guy's 0.2, we know we want to pool these 3 fractions because they're the 3 most active. And they have the highest change in slope when we run that activity assay there. So okay, so we're going to be tracking everything in terms of numbers on a Google spreadsheet, but then how are we going to track everything visually? Well, you're going to be creating a chromatogram. And so you're going to take all of these things that you're going to take the activity, the salt concentration, and the protein concentration. Those are going to be your Y's, and you're going to graph them as a function of the fraction number on the x-axis. All right. So what happens is first things first, you'll want to go in and track with your Bradford. And so we'll do that in blue, and you're going to be taking these every single fraction, and you'll see that initially you're going to have a lot of protein probably. It's going to tear off, and then you'll see more protein coming off in your gradient, and eventually it'll taper off. So go ahead and fill that in with a line there. Again, because we're measuring every single fraction for our Bradford, because it doesn't take a lot of time. Then you'll draw in the salt gradient, then you'll graph the salt gradient as well. So here's where you start going 0.2, 0.4, 0.6, 0.8, 1, 1.2. So, and again, so NaCl and molar, activity and micromole per minute or units, micrograms per microliter of protein there. So this would correspond to 0.2 molar, 0.4, 0.6, 0.8, 1, 1.2 molar. And then finally, so you can see where the protein is, you can see where the salt gradient is, but then we go in with activity. And so in theory, in our fractions, we shouldn't be seeing a lot of activity in those early fractions, and even in the parts of the salt gradient. However, once you hone in on where LDH is located, those fractions are going to contain higher activity. And if you graph it, if you run a line through, your activity measurements, you can see where the peak for where your LDH is located, and that's probably going to be located with one of the areas where there's protein, but not necessarily the most protein. And then the fractions that you're going to pool are going to be the ones that are going to be in the center and then on the sides of the top of that activity peak. And so this is the protein, this is the chromatogram that you're going to have to generate for your lab report. You're going to have to make a similar one for affinity chromatography, and you're also going to want to keep it, improve it, and then use it in your final paper. So make sure you pay attention, and if you have any questions, please ask me. All right then, and so that covers the background information that you need for this lab. If you have any we can talk more about the suggested workflow and then the list of things you'll need for your table in the period before class. In the meantime, if you have any questions, please feel free to contact me, and have a great week.